हे गाइस, मैं हूं गर्वित और अगले 5 से 8 मिनट्स में वी विल रीड replication of DNA. So guys, firstly we will discuss a very little about prokaryotes replication. So what happens? Identification of origin site, formation of bubble, formation of replication fork, and replication continues in two direction. This is what you have to remember. Okay, these are the three steps that happens in prokaryote as well as in eukaryote. So identification of origin site, that is only one in the prokaryotes, then formation of the bubble and formation of replication fork, and replication continues in two directions. Next, then the processive unbinding, which is done by the enzyme helicase. Now torsional strain, that is developed in resting DNA leading to supercoil. Okay, so supercoil is removed by topoisomerase 1 and topoisomerase 2. Single strand binding protein keeps two DNA apart and prevent their recoiling. Next, synthesizing in leading S, that is leading strain and lagging strain. So synthesize in leading strain is called as continuous manner. In continuous manner it occurs, and in lagging strain it occurs in discontinuous manner. Coming to most important points, the first one is that progressive unwinding is done by the helicase. To when the DNA is at rest, there is a supercoiling because of the torsional strain, and this supercoiling is prevented by topoisomerase 1 and topoisomerase 2. That's it. Then talking next, replication in two format. Okay, so leading strand and lagging strand. Lagging strand is called Okazaki fragments. Then the enzyme primase forms RNA primer. Okay, now there are three main DNA polymerase in prokaryotes. DNA polymerase 1, DNA polymerase 3, and DNA ligase. DNA polymerase 1 has two functions that are, it mainly do 5' to 3' exonuclease activity, that is the removal of the primer, and the polymerase activity as well. But the main role is played by the DNA polymerase 3, which plays an important, very important role in the polymerase synthesizing activity, that is from 5' to 3', and it is also required for the proofreading, that is it can remove any nucleotide in the order of 3' to 5' or 3' to 5'. Then DNA ligase, it is used for ligating. That's it for the prokaryotes. In eukaryotes there is the multiple origin sites. Replication bubbles are also multiple and the forks are also multiple, and these all occur in the bidirectional direction. Helicase and topoisomerase 1 and 2 have same role here. Single strand binding protein is replaced by the replication protein A in eukaryotes. What function SSB played was to prevent the recoiling. Okay, then leading strand needs DNA polymerase eta, which is processing the leading strand, and the lagging strand is processed by the DNA polymerase gamma. Okay, sorry, delta, that is delta, and I made it gamma, that's my mistake. Talking next, you can see here DNA alpha, it along with the primase forms RNA primer. Beta mainly repairs the DNA, and gamma is playing a role of mitochondrial DNA synthesis. Delta plays a role in the lagging strand, and eta plays a role in leading strand. Then there are two main proteins, that is the replication factor A and the polymerase proliferating cell nuclear antigen. So these two proteins play a major role. Replication factor C helps in binding of the pCNA2 strand. Okay, then RFC act as a clamping factor. So here you can see this diagram, and we will go ahead. So primase plus DNA polymerase alpha, they together forms a RNA primer, and PSNA RFC forms a ring like structure, as you can see here. Then these attachment to DNA polymerase eta and delta, and in lagging strand continuous with the delta, and the leading strand is continuous with the eta. Then this is in leading strand continuous 5' to 3', one RNA strand. Okay, in the eta 1. And in lagging strand, polymerase of nucleotides, then RNAs. H comes FEN that removes the RNA primer, then DNA ligase that ligases the DNA strand complementary one. So that's it for the replication of DNA. You can create these notes or join us on our Telegram channel. I will provide a link in description. Thank you for watching.